Well, thank you, Carla, very much, and thank you for joining us this morning. Please chat us if there are any issues if you can't hear, but I think we established that the voice is going through very well. So I really want to thank you for joining us. And what we'd like to do this morning is to give an overview of the state of 3D culture technologies and where they're going and how these can be utilized for uh, drug development and in general biological studies. And then we're going to be having a number of webinars coming up within the next few months focusing on specific technologies, more more with the focus on blood cancers, metastasis, immune oncology. So those are going to be a series of webinars based on these technologies. So as Carla mentioned, I started working on tumor microenvironment a while back, and as a faculty at Purdue for 5 years, we've been working on a number of models to really reconstruct the environment of human tissues and to better understand how the environment contributes to cancer progression, drug resistance, drug sensitivity. What can we do to overcome some of the resistance that um, is really acquired by cells interacting with their environment? So, kind of as a brief overview, we're going to go over a couple of things. We're going to touch on the conventional cell culture models and then the bulk of the webinar will focus on how we can reconstruct the 3D tissue with various aspects alluding to the complexity of the tissue environment. And then at the end, I will touch on the immuno-oncology applications of these models. And then one of the final slides will give you a nice table summarizing different technologies. So cell culture has been around for a while, and we all know we've grown cells in flasks and dishes and slides with various numbers of wells and numbers of reservoirs, different media, but the conventional methods in the end of the day give you a flat monolayer of cells. And if you look at different cell types, it's really, if you're looking at astrocytes from the brain or liver cells or cells from the vasculature, muscles, colon, skin, they look slightly different in these conventional cultures, but overall, they're still flat cells, and which looks very different from their respective tissues. So all of the tissues are three-dimensional, all of the tissues have structure to them, have multicellularity to them, which is really not recapitulated by a 2D culture. So just as a general overview, um, the cell culture paradigm really started in the 1800s with ability to maintain a medullary plate from the chicken embryo. He did it in a saline solution, and those cells survived for a couple of days, which was really a improvement over anything else that was done before. Then Eberling in 1922 was able to set up a first real cell line from a chick heart. The cell line was propagated over a number of years, and this was really the first illustration that cells can be maintained long-term in culture. And then as a first application of 3D culture, it actually came in around 1923 with Warburg looking at cellular respiration, and he was culturing slices of tissue to really look at cell respiration. Now, actually, his studies have come to light again in terms of when people are studying tumor respiration and looking at how tumor cells maintain their metabolism. So this is coming back his work. But then going forward, the first human cell line was established in 1952. That was the HeLa cells from cervical carcinoma of Henrietta Lacks. There's a really nice book published about it. And these cells have been used widely, and since then there are a lot of cell lines that have been established that are using the same methodology. And then finally, in 1980s, Minna Bissell established the importance of extracellular matrix and real tumor microenvironment three-dimensionality to the really tissue biology that is important for drug development and understanding the physiological functions of the tissue. So getting away from cell lines that are immortalized and that are not, in most cases, not very representative of human disease, Now there is a shift in paradigm towards using patient-derived material. Now to use patient-derived material, you have to have systems that can sustain this material. So really, what do we do to reconstruct this 3D culture tissue and allow the cells to be viable for extended periods of time? So again, kind of going back to tumors and tumor microenvironment, and this is an example of non-small lung cancer. This is a three-dimensional structure, which is really not captured by a 2D standard cell culture. So one of the methods of really getting this three-dimensional culture set up is cell aggregates. So one of the ways to aggregate the cells into three-dimensional clusters is to use a hanging drop model. And with the hanging drop model, cells are placed in the medium in a liquid drop, and over time, with the pull of gravity, they really aggregate into spheroids. And then these spheroids proliferate over a period of time, and the proliferation really goes for about 7 days, as shown by the Ki-67 staining here, at which point cells, really the spheroids, achieve their optimum size and the proliferation stops. So now this forms a real three-dimensional structure in a liquid droplet. To set this up, you can either do it manually and put little droplets of media in a dish, or you can use a 3D biomatrix type of plate for setup for hanging drops. Another method for aggregating cells is a levitation culture using magnetic levitation, where cells start out dispersed in the bottom of the plate. They have been attached to magnetic particles, or magnetized particles, and then when the magnet is applied, the cells levitate to the top of the culture, and then over time again form a spheroid, and depending on the shape of the magnet, the cells then can aggregate into clusters. And this is what it looks like when we have levitating cells, kind of as a monolayer, and then spheroids that are really aggregated together by the use of the magnet. And here's the device that's been used to set this up. Another model to kind of force cells into these three-dimensional aggregates is the use of polystyrene scaffolds. So this is similar to plastic dishes, but in a three-dimensional three-dimensional scaffolding with the inserts that look like these sponge-like, lava-like shapes. And then when cells are grown in it, they actually tend to aggregate. So here in the 3D is shown aggregates of cells, which do not happen when cells are just plated on plastic. They just kind of disperse. And another type of scaffold is an alginate scaffold, where cells form aggregate clusters. Now, these are not human-derived. These are not mammalian types of scaffolds. So none of the models that I just mentioned bring in any kind of microenvironment or any kind of real physiology aside from forcing cells to grow in 3 dimensions, which is the first level of complexity of a tissue. So then, to make things more physiological and more relevant, people have been looking at the extracellular matrix with, as I mentioned, Minabissel pioneering this work. So all of the cells and tissues are sitting in the extracellular matrix, which is specific for different organs. So either normal tissue or a tumor is surrounded by the extracellular matrix, and the type of extracellular matrix actually guides the behavior of the organoid. So placing mammary epithelium cells into appropriate matrix, laminin collagen 4 rich matrix, allows the formation of hollow acini. So cells start as a kind of cluster of cells, and they form a sphere. And within about 7 days, the cells within the center of the sphere die off. And those cells, then when they are resorbed, the space left by these cells allows milk production and milk protein secretion if these structures are exposed to prolactin or some other lactogenic hormones. And this is not something that can happen in the standard kind of plastic conditions. So what you can do if these cells are placed in the correct environment and exposed to EGF, they start forming ACINI. So here's one on the bottom with the hollow lumen. Now, if the same cells in the same matrix are exposed to HGF, that forces branching morphogenesis. So now you can start modulating the structures that are being formed based on what type of soluble factors are added to the culture. So one extracellular matrix allows the formation of the correct structure, and two, addition of soluble factors helps to drive generation of specific tissues. Now, having the correct extracellular matrix is absolutely crucial, as has been very elegantly shown by Minna Bissell in this study, where she placed mammary epithelial cells into the reconstructed basement membrane, again, laminin collagen 4 rich matrix, and that formed a hollow acinus with the ESA staining marking the basolateral surface and the sialomucin marking the luminal surface. Now, if you take the exact same cells and put them into a collagen 1 matrix, you end up with the inside-out acinus. So you can see that the now luminal markers are on the outside. So luminal markers are now basolateral, and basolateral markers are now in the luminal location. So having the wrong matrix completely destroys the functionality of the organoid. So this really illustrated very drastically how crucial it is to have the correct environment for the correct function. Another example comes from some of our own studies where we were looking to culture cells from the acute myelogenous leukemia, primary cells, patient-derived material, and putting these cells in plastic or in matrigel collagen 1 led to very poor survival of the cells over time. However, when we place cells into what we call an R-bone, reconstructed bone matrix, which is formulated specifically for the to recapitulate the bone marrow environment, these cells really flourished, they proliferated, and they were viable for up to 28 days, which will be shown a little bit later, and they formed these clusters in addition to hematopoietic cluster showing on the left-hand side. The right-hand side image shows stromal cells that are also growing out from this primary cell material. So this was from a bone marrow aspirate. So again, having the correct extracellular matrix allowed us to set up the correct environment and have the organ really function the way it's supposed to. And looking at the differences in extracellular matrix structure, so if you look at matrigel versus our bone reconstructed bone environment versus collagen 1, you can see that there's drastic differences between the 3D structure. This is a scanning cryo-EM imaging, so for these matrices. And then if you take the cells and place them into our bone and compare that to the in vivo marrow, so here we're looking at cell clusters, these dense spheroids, and these are cell clusters grown within either the our bone or isolated directly from the in vivo marrow, and you can see that those two are essentially identical. So again, having the correct environment allows us to reconstruct the tissue very closely to the in vivo scenario. Now, extracellular matrix is absolutely crucial and allows us to set up the correct environment. Tumors and other tissues are not composed of one cell type. So how do we really recapitulate the true environment that's composed of extracellular matrix, say tumor cells, immune components, soluble factors, vasculature? This requires more complicated models. So one illustration of the tissue complexity is shown here with figure A, panel A showing epithelial tumor in the green, and then stromal cells around it in red. And then if we look at the panels E and F, those are showing immune cell infiltration. So different tumors are going to have different extent of immune cell infiltration, which is also crucial to understanding how the drugs will function. So we really need to recapitulate this type of condition, this type of scenario. In addition to really forming the correct organoids, the tumor microenvironment is also responsible for drug resistance. So if you're just taking tumor cells and treating them with drugs, in most cases in the standard cultures, the cells will be sensitive to the therapy and will die. So this is shown on the right-hand side of this image. Now, if you place the cells into the correct environment, the interactions between the tumor cells and the extracellular matrix, between the tumor cells and stroma, between the tumor cells and soluble factors, or tumor cells and tumor cells themselves, so tumor-tumor interactions, all of those can be protective and lead to survival of the tumor, which then over time leads to accumulation of mutation and acquired resistance from the drugs, which is really exactly the scenario that happens in a patient. So now when we're testing drugs against cancers, we really have to look at the behavior of the drug within the environment, not isolated tumor cells, because those cells will then behave very differently compared to how they do in a patient. So one example of this resistance was illustrated by during our study of a couple of natural products. And these natural products are chemically and structurally similar, and we expected them to have a very similar function. But what was very interesting that we found out is that if we tested the compound parthenolide in a standard 2D culture or in a 3D culture where we use the Arbone extracellular matrix environment, but nothing else, no soluble components or any other cells that contribute to the environment, or if we used a complete Arbone system with the full complement of tumor microenvironment elements, none of those, so this is in blue, green, and red lines, looking at the viability on the y-axis over the dose response of parthenolide, really none of these conditions contributed to any significant drug resistance. And this was actually shown in patients that parthenolide works actually very well in leukemia, and it's in clinical trials for leukemia. Now, if we took the structurally similar compound engraffolide, it worked fine in 2D, it worked fine in 3D when we just looked at the extracellular matrix, again, blue and green lines. But once we added the stromal elements in the complete Arbone, engraffolide no longer functioned. So we cannot just rely on chemical structure of the compounds alone to expect them to be working in a human patient, because as you can see here, having similar structure does not really translate into being able to overcome drug resistance. Another example of this is looking at, we dissected out different elements of tumor environment and how they contribute to drug resistance. So again, we're looking at the viability of the cells in culture over the dose response of the drug treatment. And the black line shows two-dimensional conventional culture. This was primary multiple myeloma cells. This was a short-term culture because myeloma doesn't survive in 2D. And the cell viability was decreasing over time. Now, placing the cells into the extracellular matrix of the Arbone, again, primary myeloma cells, allowed us to grow these cells for 21 days. And over time, you can see treatment still had an effect, but there is a significant element of resistance now coming from the extracellular matrix. Now, when we added to the extracellular matrix the soluble environment, drug resistance increased over what is contributed by the extracellular matrix. And then finally, looking at tumor-tumor and tumor-stroma interactions within the Arbone, full Arbone environment, induced an even more significant resistance to the cells. So again, placing cells in the correct environment, one, gives you an idea whether a drug will actually function in a patient, and two, using systems that are comprehensive but versatile enough to be able to dissect out which elements of tumor environment are contributing to drug resistance will allow us to really understand what combination therapies might be useful or what type of chemistry should be added to adjust the compounds under development to really overcome drug resistance. So now some of the data that I presented really showed the need to have comprehensive models in place that would allow us to study the multicellular nature and all the other components of environment And we need these comprehensive models to really get to the clinical relevance and clinical predictivity of the drug response. So now let's talk about what models are being used now and being developed to really get to that point where we can really have an in vitro system that will be predictive of the clinical response and will track with the clinical response so that we can, one, prioritize pipelines and really go for development of the drugs that are going to be working in a patient, and two, going for the clinical applications, really being able to prioritize treatment regimens based on the sensitivity of a particular patient tumor, so going more into the personalized medicine and companion diagnostics realm. So kind of as a summary, again, we need to be able to bring in all the elements of the microenvironment, the extracellular matrix, the tumor itself, any healthy adjacent tissue, any tumor-associated stroma, immune components, vasculature, and so on and so forth. And there are different models that are being developed right now that capture various aspects of this environment. So kind of going back to the Arbonne model that I mentioned earlier, this is our platform where we're recapitulating the environment in an organ-specific manner, really reconstructing the extracellular matrix that will mimic the extracellular matrix found in a particular organ. That allows us to maintain the hematopoietic compartment, in this case of multiple myeloma, and the full stromal compartment of osteoclasts, adipocytes, osteoblasts, and stromal cells over an extended period of time. This shown 14 days, we've done it up to 28. And this was not, again, evident in the using other extracellular matrices where cells really did not survive. And here's an example of the survival of the AML cells for 28 days in the Arbonne culture, while the cells that are placed just in conventional systems die after a couple of days. Now, another model that's being used for a more comprehensive reconstruction of environment is tumor explant model. And in this system, what you do is you take a piece of tumor tissue, dissociated using usually mechanical means. It's usually just cutting small pieces, but chemical dissociation and enzymatic has been done as well to loosen up the tissue. And then a tissue fragment is placed in culture. And this tissue fragment brings with itself the microenvironment of that particular tumor. And this can be cultured over time and really getting various cells coming out. So this is the tumor fragment that we cultured from a patient with multiple myeloma. And over time, we showed kind of growing from under this tumor fragment stromal cells, and then there are adipocytes and hematopoietic cells. All of these cells grow out from the tumor, this tumor fragment. Now, the caveat here compared to some other models of tumor explants. Usually tumor explants are just placed in a culture dish with liquid medium around it, and the piece of tissue will disintegrate over time if not placed into correct environment. And in this case, what we've done is we put the cells into the Arbonne culture. So we put the tumor explant into the Arbonne culture, and now these cells, we were able to maintain these cells for extended period of time because we supplied the microenvironment, and as the tumor fragment was growing in the culture, it wouldn't deteriorate because of the lack of the scaffold and the lack of the microenvironment. So it's really important to supply this additional environment to maintain the viability of the tumor explant. Now, another new coming technology, which is really exciting, is the bioprinting technology, where cells are printed in this bio ink that includes different cell types and different materials that can be printed into a hydrogel, be it some synthetic scaffold or more, say, mammalian-derived extracellular matrix. Then these cells can be layered in specific shapes to form, in this case, a tube, for example, but you can make spheroids, you can make various shapes, you can add multiple different cell types and really reconstruct the tissue in a very controlled manner. And in this case, an endothelium, the blood vessel was reconstructed by the printing process, and there was very low cell death. There was some cell proliferation within this tissue. So bioprinting, again, has the same need for correct microenvironment similar to tissue explants, where really having microenvironment that the organoid is printed into is crucial to maintain its viability. Another new and upcoming technology is organ or lab on a chip perfusion systems, where a slide is created with multiple wells where cells can be placed, and the perfusion is applied to induce cell migration in a specific direction. So here's an example of a slide that can be used for a number of different applications where the wells that cells migrate to, you can use it for sorting or viability testing, looking at migration, differentiation, different applications. Or these individual wells can be set up to represent different organs and really reconstructing. So here's heart and bone, lungs. So really having a human on a chip type of reconstruction. And this provides dynamic perfusion with an example of a microvasculature vessel created using the microfluidic chamber with the dynamic flow. And these systems will also require correct microenvironment to set up these different organoids. Another application of our technology is really trying to recapitulate the environment of the multi-step nature of metastasis. So tumor spread really goes through a number of stages where cells go from one environment to another environment and they have to survive there. So we have to really mimic this ability of the primary tumor to break through the basement membrane, travel through the tissue into the blood vessel, and then extravasate somewhere else and survive there. And we set up a modified transwell model where we place the environment of the primary site into a transwell and the environment of the secondary site in the bottom of the multi-well plate. And the medial layer between these two environments provides the circulatory environment where the cells have to survive in an anchorage-independent state. And this is really the only model right now forcing cells into anchorage-independent state, where you can then identify truly metastatic cells, not just the migratory and invasive cells. And as an example, here we looked at breast cancer spread to the bone. And if we look at the non-malignant cells placed in the transwell, they form spheroids with the hollow lumina. They don't really migrate through the tissue, through the extracellular matrix environment. These black dots are just pores in the membrane, and there's really nothing colonizing the bone environment. If we take malignant but not metastatic breast cancer cells, they again form a spheroid, now without a lumen. There's again barely any cell migration. There is maybe a couple of cells down here and no colonization of bone. Now, if you take metastatic cells and place them in this environment, then you get cells really coming off. Right here, you can see little protrusions, and they form a monolayer on the membrane, and these are the invasive cells that have the capacity to really invade through the extracellular matrix, but not really continue on because they can't really survive in an anchorage-independent state. But the cells that colonize the bone environment down here can. And this only happens with the metastatic cells. Another use of the more comprehensive models is an air-liquid interface looking at lung cancer. And in this case, again, it's a modified transwell model where you can supply the lung-specific matrix for the cells to polarize, and when they're exposed to air, form the microvilli. And here's an example of the, apologies for of the lung epithelium just growing straight on the membrane support without lung extracellular matrix. These cells don't form tight junctions, so there's no distinct occluding staining, and there is no formation of cilia shown by this electron micrograph. But adding the coating of the lung extracellular matrix forces the formation of tight junctions and formation of cilia, which really represents now a functional organ. So here's really presented a number of different models that can recapitulate various aspects of the environment. And with the really advancement of immuno-oncology applications, it is really crucial to use a more comprehensive platform to understand how the immune compounds and CAR T cells or various inhibitors really can interact with the environment and have an effect on tumor cells. So some of the applications of the 3D models for immuno-oncology, looking at the CAR T cells, checkpoint inhibitors, different antibodies or antibody drug conjugates, and various immunomodulatory agents like lenalidomide, things like that. You really now have to incorporate both tumor and the immune components, and you have to recapitulate the infiltration of the immune cells into the microenvironment to be able to really understand the ratios of cells and how the cells interact in the activation. So one use of a lot of the models that reconstruct the extracellular matrix is simply setting up tumor spheroids in the extracellular matrix and then allowing the immune cells to migrate into the tissue and to identify the tumor spheroids. And what's interesting when we did that, so here in these little gray circles are tumor spheroids, and you can see that the immune cells, once they infiltrate the extracellular matrix, they really aggregate in the vicinity of the tumor spheroids, really going after these cells. So really having a comprehensive environment allows the immune cells to invade into the organs, extracellular matrix, invade into the tumors, and really demonstrate the function of the newly established or newly developed immuno-oncology therapeutics on a much more comprehensive human system. So to summarize the things that I've shown today here's a table just going over different technologies and their capabilities. So in terms of supporting patient-derived specimen and patient-derived material, which is absolutely crucial to the development of new drugs that will actually work in patients, two-dimensional cultures can barely do it, or very short term doesn't really work. The rest of the models that I talked about today can support patient-derived material to various degrees. So 3D scaffolds, especially synthetic scaffolds, hanging drop spheroids, cannot support these for very long periods of time, but it's an improvement over 2D scenarios. But bioprinting tumor explant, especially once added microenvironment component, can really support patient-derived material, and the reconstructed organ technologies that we've developed and supplying the specific microenvironment and specific extracellular matrices really can maintain these cells for at least 28 days. And in terms of having the organ-specific environment set up in the system, and my apologies, this 2D cultures can't actually do this. We'll correct it in the posted version of the webinar. 3D synthetic scaffolds also don't provide organ-specific environment, and neither do hanging drop spheroids or any other spheroid-type models that do not supply the extracellular matrix. The bioprinted material can provide organ-specific environment if it's added into the bioink. Tumor explants, of course, bring in their own environment, which is organ-specific, and the reconstructed organ platform as well is specifically formulated for that. Then forming native tissue organization, again, it happens to various degrees with 3D scaffolds and hanging drops. We are creating three-dimensional structures, which are not necessarily physiological, but they're 3D, they're closer to a tumor structure or an organoid compared to the 2D cultures. And then the bioprinted materials, tumor explants, and the reconstructed organ technologies can really set up a native organization of the tissue. Only the reconstructed organ technology can really mimic the multi-step progression of cancer. Microfluidic and organ on a chip technologies are getting there, but they're still not putting cells in many cases in an anchorage independent state. And therefore, this is not a true metastatic spread, but those technologies are very close and can also mimic some of the steps of tumor spread. And in evaluating drug toxicity, you can do that when you have some of the components of the microenvironment, and various models have various degrees of ability to do that. Now, in terms of scalability, 3D scaffolds, 2D cultures can be scaled for high throughput very easily. Hanging drops, bioprinting tumor explants are harder to scale, may not scale for high throughput, and the reconstructed organ can, and we're working with a number of partners to scale it up for high throughput format. And finally, in terms of the relevance to clinical response, we're at the limit of ability of 2D cultures to really provide useful information for clinical correlation, clinical relevance. Forcing cells into a 3D environment is more clinically relevant, but not very predictive. And so having really the technologies where you can have correct spatial orientation and have multiple cell types and multiple elements of the environment incorporated, these technologies are the ones that are going to be the most predictive of the clinical response. So with that, we covered all of the elements that I wanted to talk about today, kind of going from the standard 2D cultures into newly developed 3D culture models with different elements of human microenvironment incorporated. We touched upon immuno-oncology applications, which will be a subject of its own webinar, and we looked at the technology comparison. So a few upcoming webinars in September and November will be on maintenance of hematological malignancies, specifically on the reconstructed bone marrow, looking at multiple myeloma and looking at the predictive ability of the reconstructed bone platform in multiple myeloma and AML. And then in November, we would be talking about metastatic potential and how we can really recapitulate tumor spread. And then in the new year, we're going to have another webinar on immuno-oncology applications. So I really want to thank you for joining us today, and I'm happy to take questions. Also, keep in mind that both webinar and your questions and our answers will be posted on the zPREDICTA website as well as on the Cellna website. And if you'd like to contact me, here's my email. And if you're interested in any products or partnerships, please visit our website or email me.